Good morning, everyone. Good morning, everyone, and welcome to our third plenary session for ATVC 2022. First, as always, I will make some announcements for you today. The first one is to remember that our party is open to everyone. It's happening tomorrow. And be sure to get use your badge to get in, and that's it. You can come with anyone you want. And then if you're coming to our banquet, there is very important for you to have your badge with the codes on the back. That's what the codes are for. It's your banquet entrance. If you don't have the code on the back of your badge, please let us know as soon as possible so we can help you solve that. We invite you again to go to our art exhibit. It's under room 101. It's called again, No Forest, No Cumbia. And remember that Huba is a great tool for you to experience our meeting. There's a lot of tools very useful for you, and we invited you to visit our virtual posters too. Lastly, we have two special events for today that we want to share with you again. So the first one is the debate. It's happening here at 4:30 p.m. It's going to be 1 hour long. After that, we'll have a poster session for today. And our main special event for today is also the ATVC's Town Hall. It's an opportunity for you to share your experience and also plan on the strategic future of ATVCs. So you're very welcome to join us. Now we will begin with our plenary session. Today, Professor Liza Comita will share with us a presentation titled Janzen-Connell Beyond 50, Tropical Plant Enemy Interactions in a Changing World. Dr. Liza Comita is a professor of tropical forest ecology in the School of the Environment at Yale University. She's also co-director of the Yale Center for Natural Carbon Capture and Research Associate of the Smithsonian Tropical Research Institute in Panama. Dr. Comita earned a master's degree in conservation biology from the University of Pennsylvania and a PhD in plant biology from the University of Georgia. Please welcome Liza to the stage.
All right, is this on? Great. Thank you all for getting up early to come and see me. I know most of the fun of ATVC is staying out late and drinking with friends, so I appreciate you making the effort to get here so early. And I also want to say thank you to the organizers. I know it's been a long 2 years making this happen in person in Cartagena, and it's been such a pleasure. Today I'm going to talk to you about research done by my lab group over the past several years, framed in the context of the Janzen-Connell hypothesis, but really about tropical plant enemy interactions more broadly. And I want to start off by with my acknowledgements. This is not just my work. This represents the work of an amazing number of brilliant, hardworking, wonderful people, in particular students. I'm going to be talking about work by former students and postdocs in my lab, and I'll feature them as I go through, but also a huge number of people who've contributed to data collection and collaborators, and also a shout out to Christian Ziegler. Some of you may have seen his beautiful exhibit last night, but he has provided a large number of the really nice photos that are in my talk. And again, a special thank you to ATVC. It's been such an integral part of my professional life. I feel like I grew up professionally in ATVC with this wonderful supportive community. It's also played a huge role in my personal life. Some of you may know I met my husband at an ATVC final party. And so it's been a wonderful organization to be a part of for me. So I want to start off talking about something that we are all way too familiar with. So the past 2+ years, we've all been somewhat held hostage by this little virus here. And I saw this study, some of you may have seen this, it came out in PNAS last year, where some scientists calculated the total number and mass of SARS-CoV-2 virions. And they said, although each infected person carries an estimated 1 billion to 100 billion virions during peak infection, their total mass is no more than 0.1 milligrams. This curiously implies that all SARS-CoV-2 virions currently in all human hosts, and this was in mid-2021, so a lot of people were infected, they all combined have a mass of between 100 grams and 10 kilograms. And another scientist similarly calculated all the coronavirus in the world would fit inside a Coke can with plenty of room to spare. And that's kind of that hit me hard when I read that. It's breathtaking to think about how our daily lives, our societies, every aspect of it has been disrupted by this tiny little virus, all of which could fit inside of a Coke can. And then I thought about it a little bit more, and I thought, we already know this as tropical biologists. We know very, very well that the tiniest of organisms can have the biggest of impacts. And this has been recognized for a long time in ecology, particularly in tropical ecology. And I think everyone would agree the hypothesis that best captures this idea is the Janzen-Connell hypothesis. And I'm going to go ahead and assume that most people in the room are familiar with this. You heard it in introductory ecology and tropical biology courses, but I'll go over it just briefly for those of you that don't think about it all the time. So the Janzen-Connell hypothesis, or sometimes called the enemies hypothesis, was put forth back in the early 1970s. And I called my talk Janzen-Connell Beyond 50 because the hypothesis just celebrated its 50th anniversary in 2020, 2021. And it's this idea that trees produce, or plants produce seeds, but seed dispersal is limited in space. So most seeds and seedlings fall close to a parent tree, and you find fewer and fewer seeds and seedlings as you move away from that tree. And this high density of seeds and seedlings near the parent tree attracts or facilitates the spread of natural enemies, host-specific natural enemies like insect herbivores or fungal pathogens. And so seeds and seedlings occurring near their parent tree at high densities suffer from high rates of attack. And so the probability of a seed or seedling surviving close to a parent tree is low, and it increases as you move away from a parent tree. And this is a really intuitive idea when you think about it, and especially what we know now from density-dependent disease spread, it seems pretty clear. But what's really exciting about this hypothesis is Janzen and Connell went on to say, well, this could be a mechanism that explains how such high levels of tree diversity are maintained in the tropics. And this is why. So if you are a common species and your seed just manages to disperse away from you, from the parent tree, it's probably just going to bump into the next adult of the same species, the next conspecific. And so it's not going to escape those natural enemies. But if you're a rare species in the forest and you're a seed of that rare species that manages to disperse away, you're going to escape from your natural enemies. And so across the whole forest, seedlings of rare species are less likely to encounter their enemies, and this provides a rare species advantage. And that rare species advantage helps keep these rare species in the local community, prevents them from going locally extinct, and thereby helps maintain high diversity. So what I'm going to be talking to you about today is what I'm calling Janzen-Connell effects. And by that, I mean, this is a term I'm going to throw around a lot, conspecific negative density or distance dependence, CNDD for short. And it's referring to this prediction from the hypothesis of lower performance of an individual when near or surrounded by neighbors of their own species, or conspecifics. And this can occur due to strong within species competition for resources. But as we heard Lizzie talked about in her plenary on Monday, oftentimes what we know is that this is caused not by competition, but by these host-specific natural enemies. And as I mentioned before, the Janzen-Connell hypothesis recently turned 50 years old. And at last year's virtual ATBC meeting, there was a really fun and exciting session that I had the honor of moderating with Rudolfo Dirso, featuring Lissy Coley, John Terborg, and Dan Jansen himself, talking about how he came up with this idea. It was a really exciting and dynamic conversation, and I think it's still on the ATBC YouTube site, so you can go and watch it if you haven't seen it yet. So I briefly want to talk about, okay, at 50 years old, where are we? Where is this hypothesis? Well, first of all, there's been very strong support for this prediction of negative effects of conspecific neighbors. So what I'm showing you here is results from a meta-analysis that my lab group did back in 2014, where we looked specifically at experimental studies of conspecific distance and density-dependent mortality. So these are experiments where people either moved seeds or seedlings, put them close to or far from a conspecific adult or at high or low conspecific densities. And what we found, this is the effect size here. So to the left of this dotted line means a negative effect of being close to or at high densities of conspecifics. And overall, we found a significant negative effect of being near conspecifics. And in particular at the seedling stage, much more strongly and not so much at the seed stage. And more recently, Song et al. Did an updated version of this meta-analysis last year in Ecology Letters. The results were a little bit different, but largely consistent that overall the estimates tend to be negative. You see it's much stronger for seedlings than for seeds, particularly for distance. And if we focus in just on tropical trees, what we see is that these tend to be significant negative effects for seedlings of being at high density or close to conspecific adults. So there's good evidence for this prediction that tropical tree seedlings suffer when close to conspecifics. So what about, are these effects driven by natural enemies? Well, there's now been, over the last 50 years, a large number of experiments demonstrating that this pattern of conspecific negative density dependence is frequently mediated by natural enemies, and that it can promote species diversity at the seedling stage. And I'm just highlighting one study here. There's been a large number of excellent work by many of the people here at the conference, but this one is by Ravi Bagchi back in 2014 in Nature, where they used an experiment where they had control plots, and then they had, these are seedling plots, and then they had plots they sprayed with insecticide or fungicide to remove the natural enemies. And over here is the effect of negative density dependence. And when they applied fungicide in particular, they found a weakening of that effect. And they also found the diversity of recruiting seedlings was significantly lower when fungicide was applied. So this provides strong support for the idea that these dynamics are mediated by natural enemies. In this case, in particular, fungal pathogens. And there's also been, in the last 50 years, a huge body of theoretical work focused on the Janzen-Connell hypothesis. And again, I'm just going to highlight one study here by Tal Levy and John Terborg and others, where they used a theoretical model. And what they showed was that enemy-mediated conspecific negative density dependence has the potential to maintain very high tree species diversity. So this hyperdiversity that we see in tropical forests could arise or could be maintained specifically by these host-specific density-dependent natural enemies. So at 50 years old, the Janzen-Connell hypothesis is looking pretty good. So studies have provided support for conspecific negative distance and density dependence in plant communities in the tropics as well as beyond, have supported the idea that these patterns are caused by natural enemies. And also theoretical studies have shown the potential of these Janzen-Connell effects to maintain tree species diversity. And so it seems like we have this amazing body of literature supporting this hypothesis. But as tropical biologists, we know the tiniest of organisms can have the biggest of impacts, but we also know that communities are complex. And I sat through a number of talks yesterday where I heard people say, and then I found every species did something different. And I think that's usually the norm in our field. We're so used to this complexity, this variation. And this is something that I want to talk to you about today in the context of the Janzen-Connell hypothesis. So what I'm going to focus on today is talking about variation in Janzen-Connell effects. And first I'm going to talk about variation among species. and touch a little bit on how and why species vary in their sensitivity to conspecific neighbor effects, how strongly they're impacted by these effects of neighbors of their own species. Next, I'm going to talk about variation within species and present some work done by one of my former PhD students looking at evidence for within species pathogen specialization. So instead of having species-specific natural enemies, genotype-specific natural enemies. And then finally, I'm going to end talking a bit about how we might expect these effects to change in a changing world, and in particular, focusing on processes around forest degradation. And across all of these areas, what I want to touch on and think about is the implications for the maintenance of tropical tree diversity. When we start looking at this variation in these Janzen-Connell effects, does it change the predictions about how these interactions maintain high levels of diversity in tropical forests? Okay, so first, looking at variation among species. And for that, I'm going to take you to Barrow, Colorado Island, just up the road in Panama across the Darien Gap. And this is an island in the middle of the Panama Canal that is about to celebrate its 100th year of research by the Smithsonian Institute. I think there's a workshop tomorrow at lunchtime to talk about how to commemorate this anniversary. And it's been a place where research has been taking place for a long time, and we know we have an amazing amount of information about the forest. In particular for trees, we know a lot because of the 50-hectare Barrow, Colorado Island forest dynamics plot. It's 1 kilometer by half a kilometer, and it was started back in the early 1980s by Steve Hubble and Robin Foster, who's right there, who's an amazing botanist that was doing the identification of trees here. And all of the saplings and trees greater than 1 centimeter in diameter have been tagged, mapped, and measured at about 5-year intervals in the 50-hectare plot since the early 1980s. And so we have this spatially explicit data set on where all of the larger individuals are. But for the first couple decades of the plot, we didn't know what was going on in the forest understory with the smaller individuals. So back in 2001, I helped establish a census of seedlings and small saplings in the 50-hectare plot. Excuse me. We put in a 1 meter by 1 meter square seedling plot every 5 meters. So about 20,000 of these 1 meter square seedling plots in which all seedlings and small saplings greater than 20 centimeters tall up to that 1 centimeter diameter cutoff of the larger tree census. All of these little seedlings got tagged, they were measured, identified to species, and their location was recorded. And combined, these two data sets are really powerful because they allow us to have spatially explicit information about seedling and adult densities. So for each seedling with a tag in these plots, we know the number of other seedlings and the identity of those seedlings within the plot, as well as the surrounding density and identity of adult neighbors, sapling and adult neighbors. And so with that, I constructed a neighborhood model of seedling survival, where for each individual seedling, I modeled that probability of survival as a function of conspecific and heterospecific density of seedling and adult neighbors. And what I'm showing you here, I did this for about 180 species for which there was sufficient sample sizes in the data set. And what I'm showing you here is a histogram. This is the strength of the neighbor effect on seedling survival of conspecific neighbors, and the number of species that experience that strength of an effect. So anything to the left of this dotted 0 line means that the seedlings of that species had a lower chance of survival if they were close to or surrounded by high densities of seedling or adult neighbors of their own species. And what you may notice is most of these estimates are less than 0. So again, we see this support for a significant negative conspecific density dependence. But the other thing that struck me looking at this was how wide the variation is among species. And it becomes even more apparent when you compare it to the effect of heterospecific neighbors, which you can see is clustered more around 0, kind of negligible effect of heterospecific, meaning of different species, and very little variation among species in how they were affected by heterospecific neighbor density. And so this really struck me. We have this wide variation among species. And again, this is an observational data set analyzed with complex models, so you always have to take it with a grain of salt, but it's very consistent with what we see coming out of other studies. So again, this is this meta-analysis last year by Song et al. And one of their main conclusions when they looked at the effect sizes across all of these different experimental studies was that their analysis reveals very large variation in response, in this case, response to conspecific density and distance. So very large variation among species. And so this seems to be a common theme, that some species are very sensitive to the density of their conspecific neighbors, and other species don't seem to care very much. So why? Well, I'm not going to spend too much time on this, but there's a couple reasons why this might be the case. And one of them relates to species life history strategies, and in particular, shade tolerance. And this is a study by McCarthy, Newman, and Kobe back from 2008, and they did an experiment in the shade house looking at the sensitivity of different tropical tree species to pathogens. So they grew seedlings in pots with soil from beneath conspecific adults and heterospecific adults. And what I'm showing you here is species shade tolerance on the x-axis, and the change in performance when seedlings are exposed to the soil microbial community of conspecifics. And they found this nice correlation showing that species that are more shade intolerant, these light demanding pioneer species, are more sensitive to soil pathogens compared to more shade tolerant species. And this lines up with a lot of work that's been done also on herbivory, like Liza Comita talked about, where we know that species tend to fall along this axis of either growing very fast in high light or growing more slowly, but being able to persist and survive in the shade. And one of the ways that one of the things you need to do to survive in the shade is defend yourself against all of these different natural enemies that occur in the forest understory. And so we looked at this with the seedling and sapling data from Barrow, Colorado Island, and sure enough, we found that faster growing species, which are these more light demanding species, were more sensitive to conspecific neighbor density than slower growing species. And we found this in this analysis specifically at the sapling and adult stages. And so what you can see is as the density of conspecific neighbor increases, their probability of survival drops off, but it drops off much more quickly for these fast growing species compared to the more shade tolerant, slower growing species. And so it makes sense that we see this wide variation among species and their sensitivity to these Janzen-Connell effects because it maps onto species life history, well-known species life history strategies. Another trend we found looking at the BCI data at the seedling stage was that rare species are more sensitive to conspecific neighbors. And this makes sense because for a species to be able to become abundant in the forest, it can't be that sensitive to its conspecific neighbors. And this matches up again with experimental work done, in this case by Scott Mangan, Stefan Schnitzer, and others, back in 2010, also from Barrow, Colorado Island. And this is another one of these soil pathogen experiments where seedlings are grown either exposed to the soil pathogen community or soil microbial community from beneath conspecific or heterospecific adults. And they similarly found that more abundant species were less sensitive to species-specific soil pathogens. So it seems that both life history strategy and species abundance are related to some of this variation, wide variation we see in species sensitivity to conspecific neighbors. But that got me thinking, well, what is the implication of this variation among species in sensitivity to conspecific neighbors? What's the implication of that for the maintenance of diversity in tropical tree communities? And I remember Kyle Harms asking me this when I, a decade ago when I presented this, and I remember sort of giving a very hand-wavy answer, and I kept hearing him in the back of my head saying, well, how is this possible? What's going on? And so I was lucky enough a few years ago to recruit a postdoc to the lab, Simon Stump, who is just finishing up. He's a theoretical ecologist. He was just finishing up his PhD with Peter Chesson, and he was interested in the Janzen-Connell hypothesis. And it led to a really productive and exciting collaboration that we had over several years, where we explored this variation, used theoretical models to explore the implications of this variation for the maintenance of tropical tree diversity. And what I'm showing you here is a sort of cartoon of his model. And so in this case, we have 3 different species, the red, the blue, and the yellow. And they produce seeds, and a certain proportion of those seeds are dispersed away from the parent plant. And then he simulates these conspecific effects, these negative conspecific effects. So seedlings that are falling beneath adults of their own species are more likely to die than seedlings that fall of that species that are underneath adults of different species. So he simulates that conspecific negative density dependence, then an adult tree dies, and that gap, that space is filled in through lottery competition by the remaining seedlings that made it through that density dependent filter. And he simulates this for multiple generations and looks at how many species are able to persist in the system. So he did one version of this model where each species, the strength of the negative impact of growing beneath an adult of your own species is the same for all species in the system. And then he compared that to a case where you have variation in how negative those impacts are. So in this case, I don't know how well, can people see the slides pretty well? I don't have my glasses on, so I can't tell. Okay, good. So in the case of the yellow species here, what you can see is there's 3 yellow seedlings. All of them that fell under this yellow adult die. It's really bad to be underneath your own adult if you're a yellow species. And the ones that manage to disperse away, they survive. And so this species experiences very strong negative effects of conspecifics. And if you compare that to this red species, you can see it has also 3 seedlings underneath there, but only one of those 3 die. It's still worse to be under your parent because the red ones that manage to disperse away survive, but you still have a shot at surviving underneath your parent. So we incorporated this variation in how sensitive seedlings are to their conspecific adults. And then we looked at how that affected the community when you run this model over many generations. And what we found, interestingly, was that variation among species in the strength of conspecific density dependence makes communities less stable. So in other words, when species vary in their sensitivity to conspecific neighbors, fewer species are able to coexist in the system. So in this graph here, this is the number of persisting species over time in the model. And in this case, there were 15 species to start with. And in this case, this is the variation among species in the strength of conspecific density dependence. So 0 means that all species experience the same level of conspecific density dependence. And when that's the case, they all are able to coexist and persist. So these Janzen-Connell effects do maintain that high diversity. But when you keep the mean level of conspecific density dependence the same, but you allow it to vary among species, you very quickly start to lose species from the system. And in some cases, what we found was that having this highly variable conspecific density dependence can actually decrease rather than increase tree diversity compared to a system without any conspecific density dependence. And this is really shocking in some ways because oftentimes when we find these conspecific density dependent effects, you do an experiment or you do an analysis and you say, oh, seedlings are more sensitive to neighbors that are of their own species than another species. This supports the Janzen-Connell effect or the Janzen-Connell hypothesis, and therefore natural enemies and conspecific negative density dependence are what maintains the high diversity we see in tropical tree communities. But what we found is, well, if there's really wide variation among species, that may not be the case. It may not be helping maintain diversity. Unless, so one thing that occurred to us, so why do you have all these rare species that are experiencing strong density dependence persisting in the community? Well, maybe there's a trade-off that could compensate, that would allow species that are very sensitive to conspecifics to persist in the community. And the first thing we thought of was dispersal. And what I didn't tell you before was what these different lines are. And these different lines correspond to the proportion of seeds that disperse away from a parent plant. So this line over here, in this case, 90% of the seeds, and this was the same in this model, the same for all species, 90% of the seeds dispersed away from their parent plant. And when that was the case, we were able to maintain high diversity with larger variation in CNDD. But when in this line over at this end, only 60% of seeds dispersed away from a parent plant. And in this case, you started to lose diversity quickly when you had variation in these conspecific effects. So we started to think, okay, well, dispersal is a really important thing to look at. And there's been a number of other studies, including some really exciting recent ones, looking at dispersal, patterns of dispersal, and how that interacts with Janzen-Connell effects. And so Simon created a second model where he allowed dispersal to vary among species. So not just how sensitive species are to conspecific neighbors, but also how well they disperse. And so high seed dispersal is advantageous in the Janzen-Connell model because it allows seedlings to escape from their natural enemies. And species with low seed dispersal are at a disadvantage because if the Janzen-Connell hypothesis works because rare species can escape their natural enemies, if most of those seeds just fall underneath the parent plant, they're not going to escape their natural enemies even if they are rare. But what we showed is, at least in this theoretical model, is if species with low dispersal are less susceptible to specialist natural enemies, then species can coexist. So in other words, if we have this dispersal susceptibility trade-off, this could explain why species that experience strong conspecific negative density dependence are able to persist in the community. So if you can get away from your natural enemies, it doesn't matter if you're very sensitive to them. But if you are really not strongly impacted by your host-specific natural enemies, you're well defended, you can regrow tissue, then it doesn't matter if you fall close to your parent plant because you can still survive. And so again, this is an idea we've only explored theoretically, but it's an interesting thing to think about of how we could have this wide variation among species in their sensitivity to conspecific neighbors and still have these species persisting. So just quickly to sum up from this section, because I know it was a lot, what we've found and others have found is that tropical tree species vary widely in their sensitivity to conspecific neighbors. And this variation may decrease the ability of Janzen-Connell effects to maintain high plant species diversity. And I want to stress that we definitely need more work to understand the underlying mechanism and the potential trade-offs that are at play here. So for those of you that are students or postdocs and are super excited about the Janzen-Connell hypothesis, rest assured it hasn't all been done. There's plenty more to do in a lot of really exciting areas. So next, I want to move on to talking about within species variation in Janzen-Connell effects and talk to you about this study done by a former PhD student of mine, Janelle Eck, who you can see here. And she had previously worked in an agricultural extension agency. And she said, well, we know really well from agriculture that pathogens tend to be genotype specific. We think a lot about this in agriculture, but nobody talks about it very much in ecology, and especially in the context of the Janzen-Connell hypothesis. So she wanted to know whether soil microbes specialize at the within species level in tropical forests. And she set up a shade house experiment on Barrow, Colorado Island with one species, Virola surinamensis. Of course, she tried to do it for 4 or 5 and none of the seeds germinated and the seedlings died. And you all know the story, but she really got great sample sizes for this one species. And she collected seeds from beneath 11 reproductive female trees. This is a dioecious species. And she grew those seedlings in pots that contained soil that had been inoculated with soil collected from beneath either the seedling's mother tree or another reproductive adult of the same species. So we called this the mother versus other experiment. So all the seedlings were growing in soil from beneath Virola surinamensis, but either from soil collected beneath their mother or a different individual of that same species. And interestingly, what she found is that at the end of the experiment, when she looked at overall biomass, seedlings had lower growth in the soil microbial community of their mothers. And this is the overall result. This is just showing it broken down by each of those 11 trees to show it was a fairly general trend. It wasn't just one tree. And what we found was this lower growth when that soil microbial community came from the mother tree. And we also looked at soil nutrients as well as colonization by mycorrhizal fungi, and neither of those explain these performance differences. And so this suggests that highly specialized pathogens were the mechanism reducing seedling performance in maternal soils. And this was really interesting to think about. If we have these, this specialization within species, for example, on different genotypes, well, again, what is the implication of that for the maintenance of diversity? And luckily, Simon Stump was still in the lab, had just made all these beautiful models looking at variation among species. And we said, can you tweak your models and look at variation within species? And tell us what are the implications for the maintenance of tree species diversity if pathogens are specialized at the genotype level? And I don't want to go too long, so I'm going to go through this quickly, but what he found was there is a difference in terms of the effects of genotype versus species-specific pathogens, and it depended on whether species were rare or common. So for rare species, the benefit of escape from pathogens is similar regardless of whether the pathogens are species or genotype specific. And so there's this really nice image that came from a commentary by Halger Rogers and M. Frick on this paper. And this is showing for rare species here, when pathogens are species specific, seeds that disperse away from a parent plant are largely going to fall in enemy-free space and they're going to escape. And the same is true if you are talking about genotype specific pathogens. Rare species disperse away from a parent, they still escape natural enemies. But for common species, they benefit much more when pathogens are genotype specific. And that's because all of a sudden, a larger proportion of space is free from their specialized natural enemies, especially when the genetic diversity of that species is high. So if you look down here for common species, if pathogens are species specific, seeds that disperse away from a parent plant bump into these conspecifics and their natural enemies and have a low chance of surviving. But if pathogens are genotype specific, some of the seeds are going to disperse near a conspecific that's genetically similar and get attacked by those genotype specific natural enemies. But all of a sudden, all of this space opens up where they can escape these genotype specific pathogens, even though that species is abundant across the landscape. And so the implications of that for the maintenance of tropical tree diversity is that genotype specific pathogens can promote plant species coexistence, but they tend to do so less strongly than if the pathogens are specialized at the species level. And this reduces the rare species advantage and weakens this stabilizing effect of specialized natural enemies, so their ability to maintain high diversity. And I don't have time to talk about this much, but a really interesting side note from this study is Simon also simulated selection in one version of the model and found that over evolutionary time, genotype-specific pathogens will select for increased seed dispersal, so away from a parent plant relative to species-specific pathogens. And this is really intriguing because, as I talked about before, if species are able to disperse well, it doesn't matter if they're sensitive to their natural enemies. So if you have this increased selection for dispersal over time, it actually strengthens the ability of Janzen-Connell effects to maintain high diversity. All right. Finally, I want to end talking about how these Janzen-Connell effects might be changing in a changing world. And we know that forests in the tropics, ecological communities everywhere are being impacted negatively through climate change, through fragmentation, through degradation. And while a lot of the work has focused on how changes in abiotic conditions affect individuals and their physiology, what we know less about is how those changes affect important interactions. And so for this, I'm going to focus on forest fragmentation and highlight work done by another former student, Meghna Krishnadas, who's now faculty in India. And she wanted to look at how these plant-enemy interactions function in a human-modified landscape, specifically in the Western Ghats biodiversity hotspot in southern India. And she worked in this fragmented forest in a forest T matrix. So what you see here are forest fragments, and these light green squares are T fields. And she asked, do these edge effects, this being close to the edge of the forest, alter the ecological interactions that maintain tree species diversity? And so essentially what she did was she set up a series of transects like this, replicated across the landscape multiple times. And at increasing distance from the forest edge, she put in seed traps and seedling plots from about 0 to 5 meters up to 90 to 100 meters away from the forest edge. And first, she looked at the diversity of seeds and seedlings arriving at these different distances. And so what I'm showing you here is the diversity of seeds, and this is 0 to 5 meters from the edge, 20 to 30, 50 to 60, 90 to 100. And what she found was that the diversity of seeds was not significantly different at these different distances from edge. And if anything, it tended to go down as you moved into the forest interior. But then when she looked at the diversity of seedlings recruited into seedling plots right next to these seed traps, she found that from the seed to the seedling stage, diversity dropped at these sites that were within 60 meters of the forest edge, but not at these sites that were 90 to 100 meters from the edge. At that distance, the diversity was maintained at the seedling stage relative to the seed stage. And so to figure out what was going on and whether it was related to natural enemies, she set up an enemy exclusion experiment where, in addition to control seedling plots, she had plots that were sprayed with fungicides or insecticides or a combination of both, and again, replicated across this landscape. It was really an immense amount of work and really impressive. And what she found was that seedling diversity decreased with pesticides, but only in interior forests. So here I'm showing this is plots at the edge and all the way in the interior. And the black squares are the control plots showing seedling diversity. And at sites 0 to 60 meters, applying insecticide or fungicide had no effect on seedling diversity. But at these sites 90 to 100 meters in, where seedling diversity was very high in the control plots, when she applied fungicides and insecticides, that diversity dropped. And so what that tells us is this diversifying effect of natural enemies has been lost near forest edges. It's maintained about 90 to 100 meters in, she's seeing it, but it's been lost close to forest edges, perhaps due to changes in humidity or soil moisture, or to pesticide drift from agricultural fields. And this is really, really depressing to think about when we think about the global magnitude of forest fragmentation. This is a study from Haddad back in 2015, and we found that 70% of remaining forest in the world is within 1 kilometer of the forest edge. But if we focus in on this scale of the study I just talked about, 0 to 100 meters, about 20% of remaining forest is within 100 meters of an edge. And so this shift, this impact on natural enemy interactions within 100 meters could actually be very significant globally, given how fragmented forests are today. So in conclusion, over the past 50 years, we've seen that predictions of the Janzen-Connell hypothesis have largely been supported. There's a strong body of literature supporting that. But as I've shown you, there's also wide variation in Janzen-Connell effects, both within and among species. And this may reduce the ability of natural enemies to maintain tropical tree diversity. But I think it's still pretty clear that these plant-enemy interactions undoubtedly shape tropical forests in multiple ways over evolutionary and ecological timescales. And as I said before, if you're interested in this, don't worry, there is plenty more research that we need to understand how these interactions play out and what they mean for the maintenance of diversity. And finally, I would stress that we need to consider natural enemy impacts when we think about global change, not just about the physiological responses or tolerance to drought or heat, but how it changes these interactions. And I'll end with this quote from the famous paper, The Empty Forest, by Redford in 1992, that was focused on the loss of large vertebrates in forest ecosystems. We must not let a forest full of trees fool us into believing that all is well. An empty forest is a doomed forest. And I would argue that this is also true if we are losing those tiniest of organisms and those key interactions that play a role in shaping tropical forests. So with that, I'd like to thank you for your attention. And I think I have time for a few questions.
Great, so we have time for a few questions. If you could raise your hand if you had one.
Thanks, Liza, for the fascinating talk. I'm always in doubt a bit. You showed that density dependence is especially strong for pioneers, and those are rare. So it's especially strong for rare shape-tolerant species. They are rare. So how important is it, after all, to keep those dominant species in check?
So how important is our natural enemies in keeping dominant species in check? I think that's a really good question. I think it still is important overall. It's not, we don't see common species experiencing no density dependence. Often we do see it. So I think it does play a role in keeping them from becoming even more abundant, but they also may be limited by other factors. And when I think about kind of what drives commonness and rarity in general in species abundance, it's like, to be rare, there's a million different ways you could be rare, right? You could be specialized on some really small resource, or you could just be a poor competitor. But to be abundant, really common, you have to do everything well. And there are very few species that are able to do everything well, right? Like, we know that that just doesn't happen. So I think in terms of driving patterns of abundance, I would say natural enemies are certainly only one small part of it, explains a small part of the variation, but there's definitely other factors that keep more abundant species in check as well.
Yeah, hi Lisa, thank you for your talk. I was wondering if you've considered that this variation in the density dependence effects you're seeing are related to the ability of species to change their chemical profiles when they are seedlings in comparison when they are adults, because we know these ontogenetic trajectories in plant defenses vary across species. So maybe some species are able to completely change their chemical profiles when they are small, whereas others don't. So have you considered that?
I think I would defer to Lissy Coley on this one, but I think somebody asked a very similar question, and Lissy had said that they didn't find a big change over ontogeny, or like within species in terms of their defense. I know that, for example, younger leaves tend to be better defended in terms of herbivore defenses. In terms of pathogens and what's going on below ground, we know a lot less. I think for yeah, I'm not going to speculate more, but I think that, seedlings are particularly vulnerable just because oftentimes they're in the shaded understory, they have limited resources, and when they do lose tissue, they're not able to recover from that loss.
Thank you very much for such a wonderful talk. And I'm not sure I understood one part of an experiment you were showing about the role of mycorrhiza. But I was wondering if you know, like how much of the variation that you are finding among species in relationship to the density dependence and the distance to the trees, to the parent trees or mother trees, could be explained by what Susan Simard found in terms of all this supply of resources and information.
There's definitely been a lot of really interesting work on mycorrhizal fungi and whether they cause reverse Janzen-Connell effects, and in particular, ectomycorrhizal fungi seem to be better protected from pathogens and have more of a benefit of being close to parent plants. Whether that's due to some sort of resource shift from adult trees to seedlings through shared mycorrhizal networks. I don't think we know that for tropical trees. If somebody here does know that, I keep asking and I haven't heard anything. It's mostly been found in temperate and boreal species. But it does appear that mycorrhizal interactions definitely affect the strength of these negative conspecific effects and sometimes shifts them to being positive. So a lot of times where you have in Africa or South America, where you have these monodominant stands of a single species, they tend to be ectomycorrhizal and they tend to not suffer these same negative conspecific effects. So mycorrhizae are definitely an important part of this story.
Thank you very much, Liza. There's a lot of wonderful research there and you're pushing back the frontiers. I've been wondering about something that you mentioned right at the end, and that is what role Janzen-Connell may have in secondary succession. And the general pattern for secondary succession is the biomass recovers relatively quickly. The species diversity follows that, but the composition may, well, as far as we know, it doesn't recover very fast at all. And so a piece of ground that's been plowed and profoundly disturbed will lose practically all of these host-specific mortality factors. And so I just wonder what thoughts you have about the future of tropical forests, which will be mainly secondary forests.
That's a great question and something we're trying to get funding for actively right now, is to look at how this changes with forest age and recovering secondary forests. And I can see it going either way. And I said kind of the same thing years ago to Jim Beaver, who works in grasslands and thinks about plant soil feedbacks a lot. And I said, oh, well, the enemies wouldn't be there. And he said, you're talking about pathogens, whatever's, and long-lived tree species, whatever is there will, in the lifetime of that tree, adapt to specialize on that tree species. And so it is possible that those interactions could come back quite quickly. In terms of, because of the fast generation times and selection on the whatever pathogen communities are there, or it could take a while and affect the rate and the trajectory of forest succession. And so I think that's a real open question and one that I'm very eager to get data on, and that I know a number of other people here have been looking at and interested in. But I'm not completely I feel like it's not completely hopeless that we're totally losing these interactions in secondary forests.
Thank you, Liza, for a really stimulating talk. I think this is the last question that's been allowed for today. I was just wondering if you could comment on the fact that perhaps too much attention has been given to trying to prove that the Janzen-Connell hypothesis works for most or in the entire community of species, when if you think about it in terms of the spacing mechanism and preventing competitive exclusion, it's really the biggest trees that are really driving this hypothesis and its predictions. I mean, do we really care that a species with a crown that's 2 meters wide doesn't show strong Janzen-Connell effects, when really we should be focusing on trees with crowns that are 30 meters wide, and how important the spacing is, and how natural enemies prevent recruitment under those trees. So I guess what I'm trying to say is that it's fine that the Janzen-Connell hypothesis does not work for several species or even a high proportion of species, but really it needs to work for the biggest trees that dominate most of the habitat space in a tropical forest community.
Yeah, that's a really good point. One thing related to that is we tend to find that trees, and I haven't looked at it explicitly with like maximum size, but now that you mention it, I'm going to, that trees have experienced stronger CNDD than treelets or shrubs. So we know that the larger plants in the forest tend to be more sensitive to conspecific neighbor effects. And so maybe in that way, it's having an outsized effect on the maintenance of diversity if it's having the strongest impact on the largest individuals that take up the most amount of space. But that would be really interesting to look at how that maps on explicitly to maximum stature. But what I've done and what I've seen is comparisons between like shrubs and trees, and it's definitely stronger for trees.
Thank you all for your participation. We have no further time for questions, but I'm sure Liza will be so happy to answer them if you have them.